Session 13 in the first semester of B.Sc University of Mysore. The topic of the session 13 is Ultrastructure of bacteria and reproduction of bacteria. Bacteria are microscopic, unicellular prokaryotic microorganisms. The study of bacteria is called bacteriology. They are most numerous organism on Earth. And earliest fossil dates back to 2.5 billion years old. On an average, cell ranges from 0.1 micron to 1 micron. Some forms may as large as 15 microns. Structurally, bacteria are microscopic prokaryotes. No nucleus or membrane-bound organelles are found in bacteria. They contain ribosomes, a single circular chromosome in nuclear region, and the cell wall is made up of peptidoglycan. Some may have sticky coating called capsule for the attachment to host or other bacteria. Usually, one chromosome in nuclear region are present. Bacteria have additional small ring of DNA called plasmid. Bacteria are unicellular, small in size, and maybe this is measuring about 0.5 to 2 millimicron. Most grows between the best at pH of 6.5 to 7. Many acts as decomposers, recycling nutrients. Some of them they cause us diseases. This is the ultrastructure of bacteria. The bacillus type of bacteria, which shows that the outermost capsule. And morphologically, there are hair-like structures which are surrounding the bacteria, that is nothing but pili, which is made up of a chemical substance called pilin. At one end of bacteria, a tuft of long hair-like structures called flagella. Flagellas are helps in locomotion of bacteria. They are the locomotory organs. And there is a cell wall, the capsule. Behind cell wall there is a plasma membrane, organelles like ribosomes, and centrally there is accumulation or centrally concentrated nuclear material called nucleoid without any membrane. It's a prokaryotic type of genetic material. Usually, one chromosome in nucleoid region are present, and they have additional small rings of DNA called plasmids. Coming to the reproduction. Bacteria reproduces asexually by binary fission. A single chromosome replicates and then cell divides. Rapidly, all new cells are formed are identical clones of the parental type. This is the diagrammatic representation of binary fission in bacteria. A parental bacteria, bacterial chromosome, bacterial genetic material, which is going to be elongated, duplication of the chromosome takes place, and cell continue to grow. And at the center of the cell, formation of invagination, which continues to grow toward the center, connecting the center, forming a cell plate-like structure which divides the bacterium into two. Division into two cells takes place. So this is the diagrammatic structure of binary fission. Bacteria also reproduce sexually by conjugation. And bacteria can share genes by this method. Conjugation is unilateral transfer of. There is no exchange, there is no question of exchange, only unilateral transfer of genetic material takes place, during which they share the genes from one bacteria to another. From a tube-like structure between two bacteria to exchange genetic material is called conjugation tube. And these bacteria are held together by a structure called pili. Pili are short protein appendages. They are smaller than flagella. They help in sexual reproduction, adhere to the surface of bacteria, used in conjugation for exchange of genetic information, which aid in flotation by increasing buoyancy. This is the diagram of under electron microscope, the conjugation, formation of conjugation tube between two strains of bacteria, plus and minus strains of bacteria. From minus strain to plus strain, there is a transfer of genetic material only unilaterally. And there is a formation of spore in bacteria. Only one spore formed in each bacteria within the bacterium, that is nothing but endospore. Whenever the habit condition becomes harsh, unfavorable, only reserved food material is very little. And these endospores are survive and they are able to survive for long period of time as the endospores. And this is very difficult to destroy. They are heat resistant, thick walled, and many layered. Coming to the transduction and transformation. Generally, this changes the bacteria genetically. May become antibiotic resistant bacteria they are transformed. Bacteria pick up a piece of DNA from dead bacterial cell, introduce into another bacterium, and transduction takes place with the viruses. They carry foreign DNA to bacteria. Coming to the transformation separately, this is the second way in which DNA can move between bacteria through transformation. This has been discovered by Frederick Griffith in the year 1928. Transformation is uptake by a cell of a naked DNA molecule or fragment from the medium, that is the media, like nutrient agar medium, and the incorporation of this molecule into the recipient chromosome in an heritable form. When bacteria lies, they releases considerable amount of DNA into surrounding environment. These fragments may be relatively large and contain several genes. If a fragment contacts a competent cell, one able to take up DNA and be transformed, it can be bound to the cell and taken inside. So transformation frequency of very competent cell is around 10 to the power of minus 3. For most genera, when an excess of DNA is used, that is, about one cell in every thousands will take up and integrate the genes. The natural transformation which has been discovered so far only in certain gram-positive and gram-negative genera, Streptococcus, Bacillus, Thermoactinomyces, Haemophilus, Neisseria, Azotobacter, Pseudomonas, etc. The gene transfer by this process occurs in soil and marine environment and may be an important route of genetic exchange in the nature. The mechanism of transformation has been intensively studied in Streptococci pneumoniae. A competent cell binds a double-stranded DNA fragment. If the fragment is moderately large, the process is random and donor fragment compete with each other. The DNA then is cleaved by endonuclease enzyme to double-stranded fragment about 5 to 15 kilobases in size and requires energy expenditure. One strand is hydrolyzed by an envelope associated exonuclease during uptake. The other strand associates with small proteins and move through the plasma membrane. The single-stranded fragment can then align with a homologous region of genome and be integrated. Coming to the transduction. Transduction again is a type of reproduction where the bacteriophages participate in this third mode of bacterial gene transfer. And these viruses or bacteriophages have relatively simple structure in which the virus genetic material is enclosed within an outer coat composed mainly of protein. The coat protects the genome and transmits it between the host cell. Transduction is the transfer of bacterial genes by the viruses. The bacterial genes are incorporated into a phage capsid because of the errors made during the virus life cycle, and the virus containing these genes then inject them into another bacterium, completing the transfer. Therefore, transduction may be the most common mechanism for gene exchange and recombination of bacteria. There are two very different kinds of transduction, that is, generalized transduction and specialized transduction. Coming to the generalized transduction. This was discovered in the year 1951 by Zinder and Lederberg during an attempt to show that conjugation discovered several years earlier in Escherichia coli, which could occur in other bacterial species. Zinder and Lederberg were repeating the earlier experiments with Salmonella typhimurium. They found that incubation of a mixture of two multiply axotropic strains yielded prototropes at the level of about 1 in 10 to the power of 5. The generalized transduction occur during the lytic cycle of virulent and temperate bacteriophages and can transfer any part of the bacterial genome. During the assembly stage, when the viral chromosome are packed into the protein capsid, and random fragment of the particular partially degraded bacterial chromosome also may be packed by mistake. The resulting viral particle often injects the DNA into other bacterial cell but does not initiate a lytic cycle. This phase is known as generalized transducing phase or generalized transducing particle and is simply a carrier of genetic information from the original bacterium to another bacterial cell. Whereas specialized transduction, this is also called restricted transduction. The transducing particles carries only specific portion of the bacterial genome and is made possibly by an error in the lysogenic cycle. When a prophage is induced to leave the host chromosome, excision in some time carried out improperly. The resulting phage genome contains portion of the bacterial chromosome, about 5 to 10% of the bacterial DNA next to the integration site. A transducing phage genome usually is defective and lacks some part of its attachment site, and the transducing particle will inject bacterial genes into another bacterium, even though the defective phage cannot reproduce without the assistance. The bacterial genes may become stably incorporated under the proper circumstances. The best studied example of specialized transduction is the lambda phage. Coming to the importance, this is important to study bacterial ultrastructure in order to understand the cell wall structure component organelle to compare with eukaryotic cell and to understand the evolutionary significance. The basic type of cell division and sexual reproduction, which is a basic type of sexual reproduction, which is basic unilateral conjugation and transfer of genetic material, which plays important significant role in genetic engineering. Therefore, we can conclude that the actual transfer of genetic material between bacteria usually takes place in one of the three ways: the direct transfer between two bacteria temporarily in physical contact, that is conjugation, or transformation, that is transfer of naked DNA, or transport of bacterial DNA by the viruses or bacteriophages, that is nothing but called as transduction.